The title of tonight's lecture, Mosquito Control, Evolving Strategies Against a Global Threat. Our speaker tonight is Dr. Mary Kefi, a postdoctoral fellow at Johns Hopkins Bloomberg School of Public Health. Mosquitoes are the deadliest animals worldwide. Malaria alone, transmitted by mosquitoes of the Anopheles genus, causes approximately 600,000 deaths annually. That's more than any other animals combined on the face of the earth. And by the way, my students who are listening and Dr. Berlin's students, I want to interject real quick. Please take some notes so you can write a little summary for us afterwards, okay? That's important. You have to write a little summary, so make sure you take good notes. Okay, let me continue then. So in the absence of effective vaccines for most mosquito-borne diseases, control efforts focus on targeting the mosquitoes themselves. However, these efforts face significant challenges due to insecticide resistance and the mosquito's adaptability. Current research is therefore exploring multiple strategies overcoming resistance to chemical insecticides, developing environmentally friendly biopesticides, and implementing alternative approaches such as releasing transgenic mosquitoes incapable of transmitting disease. These integrated strategies aim to provide sustainable solutions to a persistent global health threat. So, oops, I lost my, bear with me for 1 minute, folks. Okay. A little short paragraph here about our speaker tonight. Dr. Kefi is a molecular biologist currently completing her postdoc training with the Demopolis group at the Johns Hopkins University Bloomberg School of Public Health in Baltimore. Her undergraduate, master's, and PhD studies were conducted at the University of Crete in Greece, where she was first introduced and trained in vector-borne diseases and malaria. She's contributed to multiple projects in the field of malaria and Anopheles mosquitoes, including the development of diagnostic tools for enhanced vector control, understanding the molecular basis of insecticide resistance, and studying mosquito parasite interactions. She enjoys studying, writing, and conducting bench work and has become increasingly interested in supporting science communication strategies. So, with that, I'm going to welcome Dr. Kefi and thank you for joining us tonight. And I will turn the floor over to you.
Thank you, Professor, for the invitation and the very kind introduction.
Absolutely.
Thanks everybody for connecting tonight. I know it's a Friday evening, but thank you for being here. So, as Professor Boris-Kivudakis mentioned, I am a postdoctoral researcher in the group of Professor Dimopoulos, and today I'm going to be talking to you about mosquitoes and strategies to tackle them with a perspective to tackle the global health that they consist. So basically, you have seen that. This is the graphical abstract of my talk. So again, mosquitoes as disease vectors. We will talk about them and their ability to transmit pathogens to the humans, and some strategies to tackle this global threat. So let's start with this very interesting, to my point of view, figure. Mosquitoes are the deadliest animals on our planet. So this is in relation to human deaths. So if you can see, I think you can appreciate that 725,000 deaths annually are caused by mosquitoes because they transmit diseases. And the cases also are, the number of cases is a massive number as well. It's 350 to 400 annual cases of cases of those diseases that I mentioned. And the next animal, interestingly, killing more humans is the human. Probably these numbers has changed the last years because this figure, I think, was prepared some years ago. But if you can see in the last place, it's the shark. When we think of a shark, we think of some animal that is pretty dangerous, right? But only 10 deaths annually are caused by sharks, as opposed to the massive number of mosquitoes. So, having said that, mosquitoes at the top of the list, with not even all of them being able to transmit diseases. So the main contributors to the disease transmission are some specific species of mosquitoes belonging to 3 different genera. We have the Anopheles mosquitoes, which transmit malaria and some filarial parasites. The Aedes mosquitoes, which you might have heard them as tiger mosquitoes because of these characteristic stripes that they have, and the Culex mosquitoes. And the 2 latter mainly cause diseases because they transmit arboviruses. So here you can see a map with the global distribution of mosquitoes belonging to these 3 genera and are of epidemiological importance to humans. And also the diseases they're transmitting, you can see them now. As you can appreciate, at least one of them, at least one mosquito genus can be found to almost everywhere in the globe. And there are several tropical and subtropical places where all of them coexist. So this graph basically doesn't mean that you also have this disease transmission in all of these places, but you have the vectors, you have the mosquito vectors there. And basically endemic for many or all of those diseases are countries in the Africa, Latin America, and the tropical regions of Asia, while also new cases are currently reported. Italy, for example, and Mediterranean region and Europe, other countries of Europe are reporting arboviruses, which is something quite new and dangerous. So people are in the scientific field, are seeking for new strategies to tackle mosquitoes. So I didn't mention the viruses here and the diseases, but I think you have heard the dengue virus, chikungunya, West Nile viruses, Zika, yellow fever, all these are arboviruses. And as we mentioned, Anopheles mosquitoes can transmit malaria, one virus, onion, and lymphatic filariasis is transmitted by all of these mosquitoes. So we'll focus on malaria here because it's what I am also interested in and I have been working on since my undergrad thesis. So I'm more comfortable to talking about them and giving you some of the data I or some colleagues of mine have produced. So malaria is the deadliest mosquito-borne infectious disease. It is owed to Plasmodium parasites, which are transmitted to humans via infective bites by female mosquitoes of the Anopheles genus, as we mentioned. So only female mosquitoes can bite and transmit. Anopheles gambiae and coluccia in particular are major Anopheles malaria vectors in Africa. And why we focus on Africa here? Because it carries the vast malaria burden. So numbers of cases and deaths annually indicate that malaria is a severe infectious disease, with Africa carrying the highest burden. From the 263 million global malaria cases and 508,000 malaria deaths, 94 and 95% of them respectively occurred in the Africa. And it should be noted that 75% of these deaths occur, unfortunately, among children under 5 years. So I believe you agree with me that these numbers are great on an annual basis. And in spite of the fact that these numbers are great, malaria cases and deaths have been averted during the first 2 decades of this century. So we had a limitation. We used to have more cases and deaths annually. And why this happened? It happened because of the use of chemical insecticides and vector control through the use of these insecticides in mosquito bed nets. So we have impregnated bed nets with insecticide that are used in households and indoor residual spraying. So we have the spraying of the walls of the houses. And this exploits the night binding and resting behavior of adult mosquitoes that like to bite at night indoors. At least the Anopheles mosquitoes that we're talking about. And pyrethroid insecticides mostly have been used in those technologies extensively. So this is why these cases were averted in the beginning of this century. However, as you can see, until 2015, there was a steady decrease on an annual basis to malaria cases and deaths. And this is, however, this declined. This decrease actually stopped and has held until now. And this is because of insecticide resistance that was developed by the mosquitoes in response to these to the insecticides that are toxic for them. So the extensive use of insecticide has led to severe insecticide resistant levels. And here you can see Africa again. So resistance in malaria vectors has been detected to pyrethroids, organochlorines. This is the class of insecticides where DDT belongs, organophosphates and carbamates. And especially for pyrethroids, the heavy reliance on this specific insecticide class exclusively in bed nets and to a large extent in IRS because it was considered a more safe option, resulted in a dramatic insecticide resistant occurrence. And here I'm showing you why resistance is happening actually. So it is because multiple exposures to the same insecticide can lead to increased frequency of resistant alleles that are naturally found in the population. So if you can follow me in that graph here, we have some green mosquitoes, which are the susceptible one, and some red, which are the resistant ones. So the red ones have some allele that gives them this characteristic to survive. And if we continue the exposure, these ones are gonna mate to each other, and then the population, after exposure to the same insecticide, will eventually be selected for that trait. It's gonna be a resistant population. So this is what happens. It's an inheritable trait. It happens at the population level. So what can we do to solve this problem? We can first try to understand the molecular mechanisms underlying these resistant phenotypes and try to improve the formulations that we have, because having new insecticides could also be an option, but it's a very slow process. It's hard to come up with new insecticides every now and then that target a complete different region. And also, we can develop alternative strategies that could either stand alone or complement these chemical insecticides that have been proven effective anyway. So here, I'm gonna give a little more background on insecticide resistant mechanism. And as you can see from this graph, we have 4 main insecticide resistant mechanisms. So mosquitoes have different strategies to adapt to the insecticides. And one way is the behavioral resistance. So basically, they avoid to come in contact with the insecticide. Is the least studied mechanism, but we know that there is the spatial avoidance and the temporal. So they avoid to come in contact either by shifting their feeding behavior to another time or by feeding outdoors, for example, instead of indoors. Then we have the very well studied metabolic resistance. where we have detoxification enzymes metabolizing the insecticide and excreting that out of the cell so as it cannot reach its toxic, its target and exert the toxic effect. We have the penetration resistance, where the exoskeleton has become thicker or remodeled, so the insecticide cannot pass through, or it has a slower penetration. And finally, we have the target site resistance, also very well characterized, very common. In that case, we have a mutation on the target of the insecticide. Usually the targets of the insecticides are neuronal channels. For pyrethroids specifically are the sodium channels. So the insecticide should bind there to prevent this channel for functioning. And when we have some mutation on the place that the insecticide binds, the insecticide cannot bind properly or at all. So this mosquito with that mutation is again resistant. And I want to focus here on the metabolic resistance to show you an example of how we can solve the problem. So people have studied that these alterations in detoxification enzymes that happen in the resistant mosquitoes. So the insecticide cannot be metabolized anymore. So I'm sorry, actually this is wrong what I'm writing here. I should write the insecticide is metabolized. It's the opposite, because we have a lot of copies of the same enzyme that metabolizes the insecticide. So by identifying one main enzyme family, the cytochrome P450s, that is involved in pyrethroid resistance, we can also come up with some way to block these enzymes. So people have identified chemicals that can block P450s. And here I'm showing you the successful example of piperonyl butoxide, which is a chemical that has been used to improve an insecticide formulation. So as you can see, in the case of susceptible mosquitoes, the insecticide can be metabolized to some extent because of the natural presence of these enzymes in the organism. However, in the case of resistant mosquitoes, we have many copies of that enzyme. We have an overexpression. So all the insecticide can be metabolized. When we add some chemical to that resistant population having all these copies of the enzyme and block their function, the insecticide can escape from that metabolism and go find its target into the mosquito and kill it. So we can restore the susceptibility to the insecticide. And this has been used. We have now bed nets that have PBO and pyrethroids, and they have been distributed and they have passed the field trials. So with that, after explaining these concepts, I would like to show part of my PhD work and the scientific questions we have in the lab. I conducted my master's and PhD. So to do that, I will introduce you to the world of mosquito legs. So why mosquito legs are important? Because they're the first line of defense against insecticide. As you can see in the figure on the left, based on how vector control is implemented, so we said that we are spraying surfaces basically with insecticide. So the mosquito lands there with their legs. So the first tissue that the insecticide comes in contact with are the legs, and it has to bypass to reach its neuronal targets. So the figure on the left shows mosquitoes on a net coated with dust particles, fluorescent particles, and under UV, you can see that this orange on their legs are these particles that are starting penetrating. This is 5 seconds after mosquito has landed there. So it's pretty fast. It's a hydrophobic dust particle that can just diffuse through the mosquito leg. So this is pretty much what happens with the insecticide. So what is in mosquito legs? Mosquito legs are covered by cuticle or exoskeleton. It's a matrix, non-cellular matrix, on top of the epidermis. And it has several layers. I don't think we should go in detail, but keep in mind that it has chitin, proteins, and some lipids, especially on the outermost epicuticle. There are several lipids and lipoproteins. So in the previous lab of Vonda's lab, Professor Vondas and Dr. Vasilia Balabanidou here found that some resistant populations of Anopheles from Africa and other regions had thicker or remodeled cuticles. So the insecticide could not pass through fast. So we have more time for the metabolism to act and the insecticide to kill the mosquito. And then what they found was that they showed that the penetration rate is lower, actually. They showed that using a radio-labeled insecticide, and that was a very interesting finding and led us to go deeper into that and detect even more things that could, let's say, corroborate that phenotype. When we tested, when compared the legs of susceptible and resistant mosquitoes, we saw altered gene expression. So this also was shown in the protein level. We had quantitative and qualitative differences. And also we had different lipid presence on the leg surface of the resistant mosquitoes when compared to their susceptible counterparts. And we produced two publications, and these show pretty much how mosquitoes cloak their legs to resist the insecticide. And also another data set gave us more information. So this experiment, basically, what we did is we exposed the mosquitoes to the insecticide for an hour and then tested what genes were altered. We saw a lot of differences in this induced condition. And one of them was a transporter that was found in the legs of induced mosquitoes with a pyrethroid insecticide. And we wanted to know more about that. So what we did, to study this transporter, let's start with that. One way to study genes in molecular biology is by removing them. So RNA interference technology has shown that by delivery of double-stranded RNAs, so we can target specifically genes and we can silence them. So basically we have less of the protein that they can produce. And this is how we do that in mosquitoes. We inject the mosquitoes with that double-stranded RNA, and we achieve that reduced expression. In case you wonder, we have specific equipment, nano-injectors, and we are targeting a specific intrathoracic region of the mosquito to do that. And then a mosquito survives the injection, and we can further study them. So we can ask specific questions like, oh, what about exposing these mosquitoes to insecticides now that they don't have this transporter protein, and see what will happen. So this is what we did. We silenced the mosquitoes for this transporter, and we saw when we gave insecticide that basically we restore the susceptibility. These mosquitoes without the transporter die more. And we also used fluorescent antibodies in confocal microscopy, and we found that this green line here is the transporter signal. You can also see the graph on the right showing the epidermal cells. Then the brown is the outermost cuticular layer. And then with this green signal, we can see our transporter having a polarity towards the cuticular structure. And along with other experiments, we propose that probably this transporter acts as a pump, which is localized in the epidermis of the legs and removes the insecticide back out. So basically, I like to mention that example. You can think of that like bailing out a boat taking on water. And this is what this pump does with insecticide before it can disseminate through the body. So one way that we could use this knowledge is that this transporter could be targeted by insecticidal supplements that block and restore insecticide toxicity. And this is what I want you to keep from that chapter, that the understanding insecticide resistant mechanism can facilitate us basically generate add-ons and synergies that could act combined to the insecticide and restore their toxic effect. So we can surpass the resistance somehow by that. And as I told you already, we also need to find alternative strategies. This is not enough. And we can exploit microbes, so like bacteria, viruses, and fungi, which are prolific in synthesizing diverse compounds in response to varying environmental conditions. And many of these compounds can have application in that killing insect technology. So either live organisms can be used or their compounds such as enzymes, toxins, and secondary metabolites, or combinations of them. And some of the advantages that they have, especially when we compare them to chemical insecticides, is that they're eco-friendly. So there is no risk of environmental spread and pathogenicity. They cause less harm to non-target insects. They can decompose quite fast, so they cause minimal environmental toxicity. And what's important here is that it's not easy for an organism to develop resistance against them. Why? Because we're not talking about a single compound here. We're talking about a combination of compounds, and it's harder to generate resistance when multiple targets are targeted at the same time. So that's an important feature. And what makes them very powerful is that they're more favorable. We have a more favorable regulatory environment and public acceptance here. And particularly the European Union now encourages biological and sustainable alternatives for pest control as part of the EU Green Deal, for example. So that's also important. Biochemical pesticides now are under the umbrella of biopesticides, but now we're talking about non-live crude extracts or the toxin or the specific metabolite. We're not talking about using the whole live organism. And in the context of mosquito targeting, we can exploit the sugar feeding of mosquitoes, this behavior of mosquitoes. They need sugar. It's essential part of mosquito biology. And so how can we use it? We can use it in an attract and kill strategy, provide these toxic molecules along with sugar and some scented component that will attract the mosquitoes to feed on this. And so as I mentioned, we start, let's say, from this bacterium and we culture it, and then we can get some powder out of this and add this powder on this nectar or on the sugar mill and make an attractive toxic sugar bait, as it is called. And here you can see it in that photo. Mosquitoes can basically penetrate that black membrane and feed on the toxic meal. But it can also be used for larval control, as this powder can be formulated to pellets that can be added to the water, because the larval stages of the mosquitoes reside into water. and we can target them by that way. So this is my professor, George, the professor of the group, George Dimopoulos. For a decade now, the Dimopoulos group has studied a very interesting bacterium in this context, the Chromobacterium sp. Panama, which is a soil and water associated gram-negative bacillus, and it was isolated from the midget of Aedes aegypti mosquitoes field code population. So several publications from the group, and here I just show some that concern the live bacterium. So the live bacterium has been shown to have a high pathogenic activity for mosquitoes of all the genera that I mentioned before, Aedes, Culex, and Anopheles, for adult and larval stages, and also in its live form, when residing into the mosquito, it can have antiplasmodial and antiviral effects. Importantly, in a non-live form, in a dried form, in a powder, let's say, with the way that I mentioned before, it has been shown also to be pathogenic for larvae and adults of Aedes and Anopheles. And I'll share with you more data on this most recent paper from the group, from Chinmay Tike, who is now a junior professor in the Ball State University of Indianapolis. So I was happy to participate in that work. And what happened in that work? So this non-live preparation of CSPP that was prepared had a high mosquitocidal activity when combined with the nectar meal. So when an ATSB was made out of them and mosquito fed on it, and it was tested against lab and field strains. So Chinmay collaborated here with the Diabatic group in Africa, in Burkina Faso. He went there and they tried that in the field. It worked pretty well. But as I mentioned before, in the field in Africa, you can find susceptible and mostly resistant mosquitoes. Basically, you can find susceptible mosquitoes currently. So interestingly, that when mosquitoes fed on that, resistant mosquitoes and susceptible died. It exerted its killing activity in both resistant and susceptible mosquitoes. However, in the field, you cannot really control how much the mosquitoes will eat, right? For how long they're going to stay on this attractive toxic sugar bait. If there is some other source of sugar, they might eat from both or prefer the other one. So what it was important in that paper, in that it showed that even when mosquitoes fed on sublethal doses, with doses that were not enough to kill them, this CSPP powder, basically solution now, because it's in an attractive toxic sugar bait, it synergized somehow the insecticides. So basically the resistant mosquitoes, even with sublethal doses, died. And also what was interesting is that those mosquitoes that fed on these sublethal doses of CSPP were very bad in seeking for a host. So they couldn't find a host to feed on. And when they did, they couldn't complete a good blood meal. And finally, even if they could complete that blood meal and they could take the parasite to be developed inside their organism, the parasite was inhibited by the CSPP. So this is a very good example of an efficient and eco-friendly alternative to the insecticide that can act against mosquitoes. It can directly kill them. However, if it doesn't, it can synergize with the insecticide and reduce the vector competence as well. Vector competence is a very important epidemiological parameter that is taken into account in the field. And basically, it means if this vector if this mosquito is a good vector, it can transmit or not. So that was my my talk so far about the CSPP, and we can now move to another strategy, to transgenic mosquitoes, as I promised. So this context is generating mosquitoes with some alteration in their genome and release them in the wild, in the field, with the purpose of either suppress the population or replace the population. So this is doable currently because of the advent of genome editing tools. So genetics have advanced a lot since CRISPR-Cas9 was identified and further studied, and has given several options to the scientists to study and also apply their research in the field. It has multiple advantages, but I won't get into detail. You can ask me if you want. So this, in combination with our knowledge in mosquito biology, has led to the ability of conducting population suppression, the next generation of population suppression, because this is something that has been conducted in the past in the context of sterile insect technique, for example. But now we have a next generation of population suppression. So release of males that will spread genetic constructs causing infertility or lethality to the offspring. Basically, with that way, we try to eliminate a population. Our understanding of mosquito parasite interactions, on the other hand, has blossomed also the last decade, specifically after 2002, when the Anopheles gambi genome sequence became available and revolutionized basically the field of this field. So currently, we can actually generate mosquitoes that are refractory to the parasite and modify the population, replace the population by releasing male mosquitoes to the field that will spread the genetic constructs with antiparasitic or antiviral properties in the context of arboviruses. But to do that, as I mentioned, understanding mosquito-parasite interaction is vital. And let's have a look on the mosquito, on the Plasmodium life cycle into the mosquito. So a complete Plasmodium infection cycle requires both vertebrate, human, for example, and invertebrate host. These gametocytes that are formed into the human are ingested by the mosquito. In the midgut lumen, the midgut lumen is the only place where these gametocytes can be fertilized, and fertilization occurs with the subsequent formation of the other stages of zygote, first of all, which will turn into a luminokinete. So this will become, after a few hours, will become motile. It's the epithelial ookinete now, and will turn into an oocyst, which after several days of development, it will get mature, and at some point it will rupture and release these sporozoites, thousands of those in the hemolymph. And through the circulation, this should come with me, it's okay. Through the circulation, these sporozoites will eventually invade the salivary glands, and this is the time where the mosquito is considered infected. If this mosquito with the sporozoites in the salivary glands bites a human, it will probably transmit the disease to them. Before that, it cannot. So having that in mind, what is our pipeline in general to study experimentally mosquito proteins that might be implicated in the infection output? So what we do is to silence those genes with a tool that I mentioned before. We use the dsRNA technology. We inject the mosquitoes and we silence specific genes at the time. And then after having depleting that protein from the mosquito, we allow them to have a blood meal that contains the parasites. After some days, we dissect their meat guts and we can stain, we can perform specific stainings for several parasite stages, but usually we're doing at the oocyst level. And we count each oocyst. Finally, we know how many oocysts were in each midgut, and by comparing and calculating a lot of them, we can know if this gene that we depleted plays a role in infection or not. And we can do the same, of course, with generation of a transgenic mosquito line that can either lack, so we have a knockout line that doesn't have the gene that we want to study, or overexpresses it in some place. We can choose the place. We have genetic tools that allow that. And then we can, again, do the same thing as I mentioned before. So this is pretty much that I'm also doing here along with other experiments. And I have an example to show you. So when I joined the lab, there was a data set, a proteomic data set. So in that data set, one protein, cryptophrome 1, was identified in the midguts of only the infected mosquitoes. And what I've seen so far is that if I silence that gene, here you can see the experimental group with the blue. I see a very significant increase in the oocysts. Each dot here represents a midgut that I counted, and how many oocysts were there. You can see that we're hundreds of oocysts in some cases, and with the pie chart below, you can see how the percentage of mosquitoes that were infected in my experiment. And I also found a chemical in the literature that can stabilize my protein. And when I fed that chemical on the mosquitoes, I saw the opposite, a very significant decrease this time. So it's the same thing if you think of that. When we silence the gene, we have an increase. And when we stabilize the protein, we have a decrease. So what does it say? It says that this protein has a Plasmodium antagonistic function. When it's there, it blocks the parasite. And when we remove it, we see the parasite increasing inside the organism. And this pretty much, along with transgenesis, is what, again, has been done extensively the last 2 decades in the lab of George's. And here I show you what we expect. Several Plasmodium antagonists and agonists have been described by this pipeline that I just explained to you. And here I also want to acknowledge Dr. Yamei Dong, who has contributed significantly to that field, specifically for Anopheles Plasmodium interactions. And most of these, along with other work, are hers or she participates. And the lab has also conducted a lot of experiments in immune genes and revealing key players of immune pathways, and also in microbiota, in revealing the role of microbiota of mosquitoes in infections. All of this is in regard with either parasite or viral infections. So again, coming back to this mosquito, to focus now on the two body parts, the midgut and the salivary glands, that are of high importance to us when we study infections in the mosquitoes. How can we exploit that knowledge that I mentioned that we currently have and implement it in the field? So as I mentioned, we have the gametocides that are differentiated into the midgut, lumen, and then we have the ookinete that traverses, the formation of an oocyst that ruptures and releases sporozoites into the circulation, the hemolymph, and through that, the parasite can reach the epithelium, traverse it, and finally get into the lumen of salivary glands, and this is where the mosquito is infective. And to cure mosquitoes from Plasmodium, we have to block the parasite at some point during that journey. So we can do this with transgenesis, as I mentioned. And here I'm gonna show you some examples of transgenic approaches used or proposed so far to generate parasite refractory Anopheles mosquitoes. So several mosquito proteins have antiparasitic activities, as I mentioned, and one approach is to overexpress them into the midgut or into the circulation. And this has been done with also with exogenous factors, with foreign proteins with antiparasitic activity. So we overexpress something in the tissue of interest and we can block better the parasite. This has also been done in the salivary glands. And the other way is to knock out mosquito host factors, mosquito proteins that facilitate the development of the parasite into the midgut. So if this green protein here helps the parasite and I remove it, I expect the parasite not to be able to pass through the next stage. And this can be also happening in the salivary glands. So for example, here, this green molecule is a receptor which recognizes the parasite, the sporozoid. And by recognizing it, it will let it in. The sporozoid will insert it. So if we remove this receptor, probably the sporozoid will not be able to be inserted in the salivary gland lumen anymore. So that's an approach. And another approach is the use of nanobodies or single chain antibodies against the parasite this time. We're not providing another protein. We instead target the parasite itself, either in the salivary glands or in the midgut. And we can do the same with antimicrobial peptides, targeting specific midgut stages, for example. So antimicrobial peptides are something like natural antibiotics produced by insects and other arthropods. And they are very effective against several microbes, also against the parasites. So we know many of them, and we can transgenically express them to target the parasite inside the mosquito. But I would also like to mention that in the context of host factors, when we remove completely these proteins, this doesn't come alone. It comes with some cost usually. Imagine removing completely some protein that is there for some reason. It does something. So sometimes we knock out these factors and the mosquito dies. It's not very fit. It doesn't want to blood feed. It doesn't lay many eggs. So we don't want that in the context of transgenic approaches to make mosquitoes refractory, because in the end, we want these mosquitoes to be able to compete with the natural populations and pass their genes through to pass these transgenes that give the feature that we want, the plasmodium resist, the feature of being resistant to the plasmodium, not letting the parasite developing inside the mosquito. So an alternative to that, knocking out completely proteins, is by transgenically expressing nanobodies or single chain antibodies in a specific tissue that can target the protein of interest instead of completely depleting that. So that's another approach. This might, I think it might be a bit too much, but let's go to more practical things like the advantages of that technology. First of all, by that we can target vectors of all spatial and temporal biting preferences. Mosquitoes that feed indoors, outdoors, day, night. And we also have access to the hard-to-target larval breeding sites. This method is specific and ecologically friendly. And with only a few releases, one can achieve a long-lasting effect. And also, when we compare it with the population suppression that I mentioned before, this population replacement strategy does not disrupt the ecological balance. So we don't eliminate the population, we instead replace it with another population. And it provides affording accessible protection to everyone without requiring human effort or behavior compliance. And why this is important, because if you think the context of vector control with chemical insecticides, bed nets are distributed in all of the household or some households, and they have to be treated with care. For instance, there is a specific way to wash them. And this is a photo I took from when I went to Cameroon with George for a field trip a couple of years ago. And as you can see, these children are playing with a bed net. They use it as a net to play soccer. What I mean is that having transgenic system not involving users could be more controlled in that sense of behavior compliance. But there are some concerns to that technology, and I think that's why it hasn't been implemented yet. So some concerns are about the predictability of the outcome, that it's something new, we don't know what will happen, and that we probably have limited capacity of remediation. One issue would be that not all parasite species will be targeted, because when we're studying mosquito parasite interactions in the lab, we're studying a specific species of parasite, and we know that each species has a different way of interacting with different mosquitoes. So this is highly possible that, of course, we will not target all mosquito parasite interactions with one transgene. We need to do more in multiple species and populations and places. That it potentially will have non-target effects, either by what will happen if, for example, some spider feeds on our mosquito, would we have a vertical gene flow or something like that. And the development of the resistance to the engineered alleles. So for these mosquitoes, we use a gene drive technology. So we can have resistance again because of mutation that will happen on the region that we are targeting. Some mosquitoes will be resistant to that, and this will spread as the context of insecticide resistance to chemical insecticide that I described in detail. Most importantly, what I believe is the reason why this is not implemented or will not be implemented at all is public consent regarding genetically modified organisms. People don't really like the term transgenic, and especially when we also mention gene drives, which is a genetic feature that allows these transgenic alleles to be spread faster. The public is quite concerned. So for this also, there's no favorable regulatory environment, and it will be a case-specific thing, I believe. So it has to do with the state, on a state level or on a continental level, I don't know. But so far, it hasn't been implemented. And for the implementation, of course, one cannot jump from the lab to the field. So if I find a molecule that really is interesting, because when I disrupt it, for example, the parasite cannot develop into the mosquito, I cannot just go and release my mosquitoes to the wild. So there is a implementation roadmap that has been proposed for that, and regulations and all of that should be taken into account. And it's a stepwise protocol to bring those products from the lab to the field, including semi-field trials that gradually increase in size, complexity, duration, while also surveillance after the release should happen. And addressing entomological and epidemiological parameters finally. So like, is our parasite-resistant population spread? Do our mosquitoes survive? Do they carry the parasite, or now we blocked somehow this interaction? Do we see, and finally the epidemiological, which is our goal, do we see less cases and deaths in that region that we implemented that? So all of this have to be done to have this implementation. And this paper that, if you want to know more about this, you can scan it. It includes a detailed list of potential hazards along with extended analysis replying to each one of them in the context of a field release population modification trial in some island. I think it's the Sao Tome and Principe, the island that they want to start this modification trial. And to do that, all these great scientists that have contributed to that field are replying to all the hazards, the potential hazards, if this is done. You can scan it if you want to know more about it. So to wrap it up, this transgenic part, this technology has not been implemented. It is envisioned mostly to be used in conjunction with existing methods for reducing pathogen transmission in disease endemic areas and epidemic situations. And also, some countries, have the goal of achieving malaria elimination, for example. This technology could be used in that context. Or, for example, if they have achieved the elimination, but now they have some reintroduction, again, this technology could be used to prevent reintroductions, but always along with some other, with other, measurements. It cannot stand easily alone. And with this, I want to end my talk here and thank all my PI, George, and all the funding sources, our insectary and parasitology core facilities, my lab mates and friends here, and also people from my previous lab in Crete, John Vondas, Vasilia, and the rest lab members, and a couple of funding sources I had during my PhD. And of course, I would like to thank all of you for your attention, and Professor Paraskevudakis for the invitation and all the organization. It was my pleasure to be here today, and I'm happy to take any questions. I'm looking at the chat.
I'm trying to find my microphone. Okay. Right. Wow. That was quite a large amount of information. My God. So many different approaches.
Yes, and there are more.
And there's more. Yeah, that's the amazing part. I was particularly interested in the Chromobacter work, and to see that, I mean, it has a lot of potential for disrupting so many different phases, using it live or using it in a powdered form. That's quite amazing that it can do all that. And does it works on viral transmission as well
Yes,
as the plasmodiums?
correct. That's
Wow,
correct.
that's amazing.
They have found some protein of dengue virus capsule that is disrupted somehow, and that's why it has also this anti-dengue effect on its life form. I'm not aware about if you feed mosquitoes on this powder, if dengue is gonna be inhibited, but I know that this happens with parasite,
Right.
with Plasmodium.
That's very interesting. And you're feeding them with these sugar-based solutions that you mentioned as well, right? That's you're adding it to that.
You basically dissolve the powder that has been dried. This comes from the bacteria, and thankfully, this powder has the toxic compound. And if you dissolve it to a solution like the sugar, 10% sucrose that we use for mosquito feeding, in specific concentrations, it acts pretty well. It kills mosquitoes.
Pretty cool. We have a question from Dr. Harold Harlan. Um, his
Yeah.
he's a medical entomologist with the
Mhm.
U.S. Army, retired now, but has a tremendous amount of experience in mosquitoes.
Mhm.
I always have to say something about Dr. Harlan because he's an exceptional individual. He asked, have you seen variable acceptance of these control methods by local human populations?
So I haven't worked close with local communities. I have a short experience in Cameroon for another reason, for a detection tool. Uh, so what I understood is that you cannot do anything without public acceptance, and you can do this by going there with some local people, some local scientists, some people who try to engage the community uh, to what you do. Because, for example, what we did, we were collecting mosquitoes from the households. So we had to enter in their house at 6 a.m. So nobody would let you in unless you have some people who have explained you and they're locals and so on. So variable acceptance, these people are using insecticides, but 20, 25 years ago, when the bed nets, let's say, were distributed, I don't know how they reacted to that. I guess someone explained them that your child and you should sleep under that because it's going to help you. And then they also saw the reduction in the malaria cases themselves and deaths, and they realized that it works. But when something new is about to be implemented, again, you have to start from scratch, right? Now, releasing mosquitoes, if you think of that, is not the more comfortable thing that one will see and will feel, you know, okay, like one will think we're trying to avoid mosquitoes, and this crazy scientist came here and released thousands of them. So I think African scientists are working on engaging the communities to be able to accept those tools and explain why. So specifically for transgenics, I already mentioned that not only in Africa, but also in our countries, I believe it would be quite difficult to get public consent. But for biopesticides, for sure, you can sell it better. It's better than insecticides. It's not that toxic. It's a natural compound. Also, mosquitoes have been released carrying Wolbachia. Wolbachia is a natural bacterium and the symbiotic to several mosquitoes, but it can disrupt mosquito populations by causing some incompatibility during the mating. And these mosquitoes have been released in Brazil, for example. So I guess the community here was engaged, was ready for that. But when it comes to transgenics, we don't really like the word transgenics. So it's more difficult.
Hmm.
So have you seen acceptance and how significant has resistance to the genetic silencing been in Philippines? So this is the resistance to the genetic silencing. You mean to the gene? Yes, to the transgenic mosquitoes, I guess. It can be a problem. People in the lab and in semi-field conditions, they do that by releasing mosquitoes in caged populations so they can monitor if resistance will happen. They have seen that this is happening, but they have come up with some different genetic tricks to fix that problem too. So most of the concerns that I mentioned here when it comes to transgenics have already been somehow solved by the scientific community already. So there is a reply to that concerns. And one of them is it concerns resistance to this. But resistance will happen to pretty much everything that we're gonna give or do to the mosquitoes. It's a response. That's why this CSPP example of multiple compounds, not just one, has a significant advantage because resistance cannot be easily cannot easily happen.
First payment.
And also to complete that reply, for population replacement, we would not only act one gene that acts against one target. The construct, the genetic construct, contains multiple of anti-parasitic genes, so as resistance will be more difficult to be developed to all of those different things. Mm-hmm. Someone wanted to ask something, and I interrupted.
You're unmuted.
That's what I could ask.
Ed, do you have a question? I don't hear any questions. Ed, if you have a question, please ask. You're muted right now, so we can't hear you.
Yeah, Ed's saying he doesn't have a question.
Okay, no problem. I was thinking about so many things when you were going through this, and now I can't even remember what they were.
Okay. Yeah, it was a
Oh, I know.
different chapter, so.
I did have a question about the bacterial metabolites that you were talking about that can be used. You mentioned there were several different types of metabolites and all that. Could you name a few of those? I think you didn't really specify which ones that
You mean
were.
in the CSPP case?
Pardon me?
In the Chromobacterium?
Well, Chromobacterium was one, but I think before you were speaking generally about different bacterial proteins. Were you referring to Chromobacter in that regard? It was the slide that came before the Chromobacter.
Yes. So the secondary metabolites, several other microbial biopesticides, basically. I will not come up with some, so I cannot recall, but several times the secondary metabolite is the one with the toxic effect. And I don't think I will recall some specific one, unfortunately. And I don't think we know many of those, but we know sometimes that it's a metabolite. So you mean somewhere here that they can be metabolites?
Before this, I think it was.
Yes.
There we go.
Yeah. For sure they can be metabolites, but I cannot recall an example right now for you. But I know in that pipeline of identifying the compound or the compounds, one should separate the protein and the protein fraction from the rest, the one that contains the small chemical molecules. And with these fractionations in that research, one can find which is the most. I can think of one. I think Harmain, for example, from Professor Lorena Lab at Hopkins, was a molecule with toxic activity against parasite from some bacterium. This was an endosymbiotic bacteria, though, for mosquitoes. I don't know if it was intended to be used in that context, but yes, people would separate these small chemicals from the rest macro molecules like proteins and toxins to see where the toxicity is in the metabolites or in the proteins, let's say, or both.
Okay. I guess there's a lot of work being done in this sort of thing too. So it reminded me of a commonly used bactericide of Bacillus thuringiensis, BT.
Yes, the PD toxin. That's a protein. That's a toxin.
Exactly. Yes. So that one has been widely used in our country.
Yes.
It's even, of course, become a transgenic component for corn.
Correct.
And it works very effectively.
That's right. And resistance has also been occurred there, but there are, again, different strategies to combat this.
Okay.
Yeah.
Very good. Well, I was it's a lot of different things to take in here,
Yes,
so I don't know.
probably it was too much information, but I tried to give a broader picture and not focus on one molecular biology-oriented project, but on the concepts mostly. I hope it was interesting after all.
Oh yeah, you have a compliment there in the chat box if you see it.
Mhm.
Um,
Thank you so much, Dr. Harold. Skip
he
of
also had
position.
a question
Yes,
about
yes.
the oviposition. I don't think we addressed that question. Do you see that one? Do you think skip oviposition may be a problem for effective disease control in cases of vector mosquitoes that often employ skip oviposition?
Um, skip of a position may be a problem. I'm not sure. I understood skip of a position again in the transgenic concept or in general. If we try to, if
Perhaps,
you mean,
Dr. Harlan, if you want to unmute your
yes,
microphone, you could explain.
yes, please, you can. You, from what I understand, if we try to make the mosquitoes skip oviposition, like if there are no breeding sites, if there is no available water for them to lay their eggs, and mosquitoes, vector mosquitoes. So I think that's not very, if this is what you mean, I think in Africa or in those field conditions that might not be so easy. It's anywhere that there is water, they can lay eggs, and I don't think it's easy to remove the breeding sites. But when we are in our garden and we just try not to leave water there because more mosquitoes will come, and we don't like that, this is more doable in a small setting, I guess.
Do you see the chat he has typed
Yes.
in?
Oh no, skipper is when female lays a few eggs in several different sites. Oh, I didn't know that term. I'm sorry about that. So, okay, in the context of transgenics, you mean? Of interesting control. So I'm not sure about that, but if I'm gonna think out loud. So if a mosquito that is resistant, for example, lays eggs in different sites, then this trait will be the trait of the resistance to insecticides will be also disseminated in another region. So clutch among several sites in succession. I see. I guess I have to think a little bit more on that, but it does sound like an extra layer of complexity because these mosquitoes can be found in different places, and either the genetic trait or the resistant alleles can be spread. In one case, this would be good, in the other could be bad. So it would have an issue, it would be a concern for vector control, but it would be a good thing if these females were transgenic. I don't know how this behavior starts from the first place. I guess it's a species-specific thing. Aedes albopix, that is okay. So it's specific to some species. I didn't know about that. Thank you. I will read about that. For Anopheles, I haven't heard. But for sure, understanding how mosquitoes are in their natural environment and things like the one you mentioned with oviposition is important to eliminate mosquitoes or to, decrease them. It's an important thing to have in mind. It's okay. My microphone does not work. I'm sorry, I didn't know that context. So
That explains something. The microphone isn't working.
just so
So
that
I was hoping that we could hear you
yeah,
speak,
he
Dr.
could
Harlan, but that,
explain us more about that. But yeah, it looks like that it's specific to some mosquitoes.
right. So, okay. Anybody else? We have any other questions? Any students want to ask any questions?
The students would usually ask good questions, but now I guess they're a bit shy.
We have some feedback there.
Sure, I can give you mine.
Any students want to ask a question or type something in the chat? I hear some voices there, but I'm not sure what we're
Yes,
hearing.
it's a question.
Shika, is that you? Are you trying to ask something, Shika? I hear you're okay. Never mind.
It's
All
okay.
right.
If any of the students would like to ask something, they can contact me to the email that I just sent to the chat, or anyone else.
Okay. Well, having said that, if there's no other questions, we have recorded this too, so it warrants another look back at it because there's so many different methodologies in
Yeah,
research work. It's pretty amazing. I was trying to take it all in, and it's quite amazing. So we'll have to actually talk about this later and see. Maybe
sure.
we can get
Anyone who
some.
wants, you can contact me.
Yeah, definitely like to see maybe another lecture on one of these other aspects, or other folks who are working there. Well,
Absolutely. There are several postdocs in the lab, but they would be happy to give talks on similar and different things today regarding skip oviposition. Okay, I see that chat was paper today regarding skip of it. Okay, that's interesting. So it also happened in Culex species. Okay, I'll definitely read about that.
okay.
Thank you.
One of my
There's
students asked a question about the female
Yeah,
mosquitoes.
I just saw it. That's a good question. So yes, females need the blood to for the eggs to be developed. So they need the nutrients, and there's for their oviposition and their egg development. So their ovaries, when we have a mosquito in the lab that is feeding on sugar and you dissect it, and when it feeds on blood, and you dissect it again, I mean, you can see that the ovaries are different in size, and that the eggs are evident there. So without Anopheles, with one blood meal, they can lay eggs multiple times. I'm sorry, with one mating and several blood meals, they can lay eggs multiple times. And they feed on blood, and 2 to 3 days later, they lay their eggs on the water. But males do not really want to, they don't need that. So unless you leave males without any other source of water, they wouldn't go on blood on the host to feed. They wouldn't touch it in the lab either, unless they're completely thirsty and this is their water sugar source kind of. But females are seeking for that with olfactory cues. They have evolved to seek for the blood meal, to smell somehow the the host and go to blood field to be able to lay their eggs.
Okay. One last thing. When we're dealing with transgenic mosquitoes, and you mentioned several different approaches to constructing a genetically modified mosquito, right?
Mhm.
So, how many generations can you expect for that gene to remain before it can be lost? Because usually they get lost after several
Yeah,
breeding, right?
there were some graph with the gene drives that I wanted to include, but it was all, I thought eventually it would be too much. When you use a gene drive, you don't have a Mendelian inheritance. You basically spread the allele faster. And this can happen. You will see that if you, this depends on how many mosquitoes you will release. In field cage trials, people have shown that after releasing, for example, a specific number of males or females, but usually they do release males because this is what they want to do finally in the field, because males do not transmit. But so my point is that depending on the initial population and the population that you're gonna release in that, assuming that you don't have overlapping generations there, because then it becomes more complex, probably it will be stable without resistance for 10, 20 generations, depending.
Wow.
Yes, but this is again in cages. When you go to the field,
It's not
I'm sure there are
quite.
some larger cage trials, but now I'm not quite sure about the number. But for sure it's gonna be more complicated in the wild. These mosquitoes have to be fit. The number of the release the initial input to the field will be very important, and how fit they will be to mate and pass the transgene to the next. That's why they also are trying to implement this in the context of a field trial in natural conditions, not in caged populations, because it's important to know something to have the reply to your question. For example, for how many generations do we expect this to be stable in the wild? And it's good not to be that stable. It's good for this to require multiple releases rather than remaining for so long. I believe it might financially it might not be so good, but for public consent, for a public consent aspect, knowing that you have something that will eventually faint rather than something that will be really stable, I think it's a good thing on one side point of view.
And just to put things into context a little bit here for the students, Mary, typically a female mosquito would lay about how many eggs in her lifetime?
Some hundreds, I guess. Again, it depends on in her lifetime. Oh, probably thousands.
Thousands.
I guess depends on how many blood meals, what species we're talking about.
Yeah, okay. But it can be thousands even, huh?
Yeah, in the lab we have done experiments for viviposition with single mosquitoes laying eggs. It can be hundreds, it can be like 20 from some hundreds or a few hundreds. So
Okay.
if we consider that based on their gonotrophic cycle, which is around 3 days, so meaning from having blood to laying eggs, if we consider the days that these mosquitoes will survive, if they have time to feed 5 times, 4 times, we might reach the number of thousands, or I don't know if it's also more.
Wow. So it's quite a huge number. So
It's
all right. Well, does anyone else have any other questions? And if not, I want to thank Dr. Kefi for
Thank you.
being here tonight and providing us with such a wonderful lecture with so much information. So thank you so much.
Thank you very much.
Appreciate
Thank you
you.
for the invitation, and all you for your attention and your nice comments here in the chat. I see them. Thank you. You can contact me if you want if I can help you with something or if you want to discuss more. Thanks a lot.
Thank you so much, right? So
Yeah, I shared my yes,
students are very happy. Thank you, students, all of you for joining us tonight.
Thank you.
Wow, lots of good
Well,
accolades there. Thank you, Rachel, and everybody else for your comments. We appreciate that. So I'll see you guys back at school then, and we can talk about this some more at the college.